Diffusion of Particles in Various Solutions by Stephanie Yorga, Maddie Dodson-Garrett, and Morgan Shipp.
The aim of this experiment was to determine the relationship between the composition of solvent and rate of diffusion of a 2 micrometer particle using a microscope and the application tracker. The variables in our experiment were: the independent one was the type of solution, so we used water, cornstarch, and soap solution. Our dependent variable was the rate of change of position over time for the various particles in each solution. So we had 5 trials per solution for a total of 15 trials. Our control variables were the seconds of data footage used per trial, so we had a total of 80 increments, and the size of the particles was kept constant at 2 micrometers. Our hypothesis was: we initially predicted that the fastest velocity would be the soap, then the water, and then finally the slowest would be the cornstarch. The slippery texture of soap and the thick texture of cornstarch led us to believe that particles would have a much easier time reaching their maximum velocity in an environment with low friction, such as soap, versus an environment with lots of blockage to pass through, such as cornstarch.
So for the results of looking at the microbeads in the water, we looked at the displacement versus the time for the microbeads, and you can also see the tracker application. So we saw that a common trend was there were random collisions, but overall the motion was in a linear pattern. And while particle 2 had the lowest overall displacement, that meant that it also had the slowest average velocity. When examining the average velocity of the different particles of microbeads in water, we found that the particles had very different average velocities. For example, particle 2 was an outlier, so it may have moved much slower because it may have encountered more collisions with the solution, causing momentum transfer, or it could have collided with another microbead, making it go much slower as it transferred its momentum. In the water, the microbeads overall moved at a slower average velocity than in the other solutions.
The results of cornstarch were similar in the sense that the motion of the particles was mostly linear. The average velocities of the particles in cornstarch were overall relatively medium to those in water and soap.
The data for the microbeads in soap show similar patterns. The displacement versus time graph was mostly linear for each particle. And in the tracker, you could tell that all the particles moved across the entire screen. After calculating the average velocity of each particle, it seemed that the particles that were in the soap solution tended to have a much higher velocity than the particles in the water or the cornstarch solutions.
In conclusion, we found that particles in cornstarch and water had similar average velocities, which were lower than those in soap.
In this experiment, we found that our expectations from the pre-lab were consistent with what we actually found, and that the motion of the particles was random. We found that the beads were moving about randomly because they were experiencing a lot of collisions with particles in the solution around them, and these movements are referred to as Brownian motion, which causes the dispersion and mixing of various substances. And we can see that this is diffusion, because when a particle collides with another, some of the momentum is transferred to the other particles so that they come away with different velocities.
So why were the velocities different? The soap solution showed the highest velocity for the microbeads, as predicted in the hypothesis. This is due to the tremendous lack of friction that occurs between soap molecules and the microbeads in the solution, allowing the beads to maintain their change in momentum from a collision as kinetic energy, rather than it being quickly converted to internal energy due to friction, like in the cornstarch solution.
We had multiple sources of error that may have affected our results.
We can also compare this experiment with a microscope to the computational simulation that we did regarding diffusion. The microbeads in this experiment represent the proteins that we saw in the computational simulation, while the water, soap, and cornstarch particles represent the water from the simulation. The varying solutions could represent different environments in which proteins exist in the body. One major difference between the two experiments was that in the computational simulation, the collisions were elastic, so the particles bounced back from a collision at the same velocity that they came into the collision at, while in the microscope experiment, the collisions were not elastic, and this meant that some of the momentum was transferred from the particles to the surrounding solution instead of all being conserved within the particles.